Welcome to PCB. Okay, second lecture. We got stuff to do. Okay, let's go through all this fluff that I tried to upset you with last time. Where were we? We were in neoteny, I believe. We were trying to eventually circle around and bring this down to molecular biology and why you should care. So I've taken sort of a roundabout way to introduce ourselves as biological creatures, the creatures resulting from genetics. We sort of are looking at how we got here, how diverse our population is, and then we're trying to draw it down to what we're beginning to know about specific genes and mutations that could have helped us along the way. And the first example of a molecular influence here that we can sort of nail down, we're very suspicious that think this might be significant, is the SIRGAP2 protein. It has duplicated 3 times in the human lineage, not so in chimps. And the first was about 3.4 million years ago, then 2.4, and then about 1 million years ago. And you'll see there are some rough correlations with the emergence of distinct hominid forms. It turns out that this gene, and here you see the ancestral one was right here, I believe, and then it made the duplications. Actually, this is the ancestral one. This is the first duplication 3.4 million years ago, and then you see the next right here, and then the last was to over here. In these duplications, the gene has been shortened, so it's not a perfect duplication, and it turns out that the gene involved here, that's not the next slide, is it? Oh, this is the one kind of out of order here. This gene, SIRGAP2, is involved in neural development, specifically in development of the brain as an organ, and it influences the rate of migration of the neurons as they go to establish the layers of the brain, and also the number of connections that are possible. So if you look at the connections, it's based on these stem neck structures. I think last time I was calling this the stem, but it's called head neck structures. They become long elongated. The neck is a little longer in the human variation. And these dendrites do not migrate as fast. And paradoxically, brain development has slowed down in Homo sapiens, prolonging the juvenile stage of life. Like dogs, they are really puppies. They never get out of their puppyhood, really. They're always eager and want to learn and always a good sport about stuff. Well, we have selected those same traits in us in the process called neoteny, prolonging the juvenile stage. It has allowed us to be flexible in our learning over many years of our lifespan, and that in turn has enabled us to develop higher levels of culture. So that's what we think is going on with the SIRGAP2 duplication. The duplication, being a truncation, can still bind. It forms a dimer when it normally operates, functions with itself. The wild type, it functions as a dimer with itself, helps brain development. But when it dimerizes with the truncated or shortened form, it's insoluble. So it really decreases the effectiveness of the SIRGAP gene expression by removing some of the active population of molecules. We have begun to inhibit SIRGAP2. We have a little bit too much, and by doing that, it has helped retard our development. We are retarded so we get smarter somehow. Okay. This is from a paper. I think this is, as you see, it's written in German. I think this is about 1886 or so, where the neoteny was recognized in apes, but it's still a valid, what the hey? A valid observation. You see it in dogs, the snout is shorter. Why is the snout related to prolonging the juvenile? It turns out that many of the genes that affect morphology of the face are just physically close together to those that affect brain development. So it's in mammals. So it's just by serendipity that if you select for cuteness, you also select for puppyhood. And we have selected for puppyhood in us, neoteny. So this, we're like the peak of the pyramid, right? We're so smart. Let's look at culture as a phenotype. Is there a limit to what we can do? Are we finished? Are we done? We can close the book on evolution as far as Homo sapien is concerned. How much of human culture is just bounded and constrained by these little mutations in our genes? So if you look at culture as we look back through the geological and archaeological record, you see the only thing that has survived are the rocks. And you see from 2.5 to 1.7 million years ago, this is Homo ergaster. He was beginning to play with rocks. But look, for 800,000 years, there was no breakthrough. It seemed like there was a constraint. Homo erectus wasn't getting any smarter. His environment apparently didn't select for smartness. He was smart enough. He was big, he was strong, and he was mean. And so if you were a little antelope or a little ground squirrel or woodchuck, you didn't stand a chance. There wasn't any reason for him to get any smart, it seemed like. How about here from 1.8 to 250,000 years ago, you have this little bit more advanced, but for 1 million years, that's it? That's all we could do? Were we just stupid? Yep, we were. We were just stupid. And 200,000 years ago, this is the beginnings of Homo sapien, and you see how we've now progressed. And 50,000 years ago, we were into pretty fancy stuff, evidence of sewing, needles, art, and iPads. When was fire developed? Way back here in the stupid stage. You going to give stupid people matches? Apparently evolution did. Here you go, boys. See what you can do with it. You have to play with that outside now. Don't bring it in the cave. Neanderthals, those charming critters. And then finally, Cro-Magnon, modern man, and all the diversity that is with us. And that cat there. Okay, so what makes us distinct from the more archaic humans? And that's a question that is driving a lot of science. One of the indications is art. There seem to be examples of body decoration in Neanderthals, a little red paint here and there, some burial perhaps, customs, but it really takes off with Cro-Magnon. These caves in France, I mean, that could go in the Louvre, right? I mean, that's modern art right there. But you know how, like most of us, the longer we stare at something, the more we see pictures and faces and all sorts of stuff and everything, whether it's floor tiles or, have you looked at those desks, those counters at McDonald's with all the polka dots everywhere? little tiny dots? Have you ever tried to see whether there's a repeating pattern? There is. It's about this far. All of those polka dots repeat. They were printed on a big drum. Oh, that's something you should figure out yourself. And now I've just given you, one of life's little tidbits there. So let's go back and look at this picture. I'm going to hit the transition button up here, and you'll see what I sort of did with Photoshop. To me, that is Cro-Magnon himself looking at me. See how the eyes beginning to darken there, and now the nose and the mouth and the lips. Suppose you were in the candlelight going through the cave. There's a whole bunch of your buddies, and you just sneaked off, and you're only like 14 years old, and you're back in the deep bowels where they do all the ceremonies, and you look up and see that guy looking back down at you. That's before. And then this is how it slowly, in the shadows, becomes Gaga. Okay, back out of the cave in the sunlight. So how did we get here? Well, we came from Africa, and you see this little place right there. That's where everybody eventually has a mama. Right there. We're all Africans. If you look at Africa today, they have the greatest genetic diversity of any other region. That's usually the hallmark of the beginnings of a species. If you look at the cow, you'll see in Northern Europe and other places, most variations of the cow are where it first was, I guess, domesticated. It's domesticated several places. But anyway, everything we say, we know, says that we were all one back in Africa, and about 1.8 million years ago, a couple of fellas, probably a whole population, it could just be a little bit each year, they just migrated just a few miles, but eventually they left Africa and they populated the rest of Eurasia, and that was Homo erectus. Homo erectus. And this is before Homo sapien. And there was a population that Homo erectus when everybody was Homo erectus, but that population that stayed back in Africa continued to adapt to the changing environmental conditions, largely of the expansion and shrinking of the Sahara Desert. That has influenced strongly the way we are now. It's caused us to sweat. Most mammals don't sweat, they pant due to persistence hunting, where you chase down an animal and just wear it plumb out. That works best in a hot environment. They just, the swiftest antelope, eventually, if you're smart, you can figure out where they're going, and you just keep trotting along behind, and they want to rest, and you keep trotting along behind until finally that poor animal's exhausted. It's called persistence hunting. We developed the ability to run long distances. We lost hair, picked up pigment to protect us from the UV, and we were adapting to dry desert conditions. But Homo erectus, not so much. He was kind of stable, didn't change much. But that population back in Africa was under severe environmental selection. In fact, until about 200,000 years ago, well, maybe 50,000 years ago, there were 3 variants of Homo sapien walking the earth. There was Neanderthal, Homo erectus, and Homo sapien. So the first branching out from Africa was by Homo erectus. But let's talk about the movement from Africa of Homo sapien, and that's on the exams and homework. You'll see the first Exodus versus the second Exodus. And I'm talking about of modern Americans, modern Homo sapien. So there was a first wave that happened somewhere between 120 to 115,000 years ago. They left by the northern route because the deserts had shrunk. You see it's kind of green up there. And they made it as far as the Mediterranean. But this first wave didn't really persist very long. There's some of that DNA in human populations, but they pretty much died out. And most of the rest of the earth was populated by a second exodus from Africa that happened quite recently. 70,000 years ago, 70 to 50,000 years ago or so. So let's back up. We left out some stuff. So we have Homo erectus, 1.8 million years ago, began to explore the earth and spread over the surface, but didn't change much from that. But the population of Homo erectus that remained in Africa was still rapidly changing. And it changed into something that we first discovered in Europe, in Heidelberg, Homo heidelbergensis. The Heidelberg form of Homo sapien actually developed in Africa also. We just first discovered his fossils in Europe. It turned out that Heidelbergensis was just before the split between Neanderthal and us. So Heidelbergensis went to Europe and became Neanderthal. He adapted to the cold. He became low and squatty. He lost a lot of pigmentation, and some of them were redheaded. Turns out the redhead genes in a gene called MC1R, mutations in that gene, if you have both of them, one from each parent, in modern humans makes you a redhead with pale skin. That gene developed in Neanderthal. But the particular mutations, the particular point mutations, are not the same as what some of the modern humans have. So we didn't directly inherit that from them, but it's just a natural adaptation to cold environments. It happened twice. But there are things that happened in Neanderthal that we have the same point mutations in us. What it seems is, as we learn more and more about the story, is Neanderthal hit the highway, left home about from 400 or 600 to 400,000 years ago. He left home as Heidelbergensis and he turned into Neanderthal. And then several other forms. Denisonian, there were several archaic, like Neanderthal creatures that were cold adapted. That happened 400 to 600,000 years ago. The Heidelbergensis that remained in Africa continued to change, but they were not adapting to cold, they were adapting to persistence hunting and the shrinking expansion of the Sahara Desert through alternating periods of glaciation. And that Heidelbergensis population, instead of developing into Neanderthal, developed into Homo sapien. And then that population that remained in Africa, when it left in two waves, which I call two exoduses. The first exodus is this not so successful one that populated the Mediterranean shores. Palestine, Gaza Strip, Mount Carmel. They found Neanderthal up there too, but more about that in a minute. Okay. As Homo sapien, especially in the second wave, let's go to the next slide, they almost go extinct. And that's where that second population, that first exodus gets sort of isolated and dies out. The numbers vary. You see it anywhere. You see it says we were down to as few as 10,000 mating pairs, Homo sapien. That's everywhere. That's up here and back in the cradle. Some estimates are 600 mating pairs. We almost went extinct. That was not too long ago, from 90 to 70,000 years ago. A big bottleneck. Then you have the second migration. And this time, we went through the southern route. And that happened 70 to 60,000 years ago. Of course, Homo sapiens spread throughout Africa and also into Eurasia. And notice what happens here. Continues to spread until it populates all of the earth and of course all the way down into South America. But notice, it begins to make contact in this region, roughly in this region here, with Neanderthal that was already there, that had already been there for 300,000 years and was fully adapted to coal. And there was interbreeding. And it seems, what the story that's emerging is, that contact with Neanderthal allowed the second expedition to now pick up traits that were cold adapted and allowed us, facilitated this time our spread to the far corners of the earth, that contact with Neanderthal. Now when we made the contact with Neanderthal, did we add genes or did we throw away some Homo sapien genes and substitute Neanderthal genes? That's what we did. How about the population that remained in Africa? If you look today, there's no evidence. They are fully Homo sapien. Uh-oh. That means everybody else is not fully human? Yes. That's what I meant when I said that some of the great figures of theology and religion and political and wars and human history are not fully Homo sapien. Most of us sitting in here are not fully Homo sapien. Some of us are, many of us are not. Oh no, wait till Mama finds out what I'm learning in college. Okay, let's get some deep breaths here. What do we need to know for the exam? Enough of this stuff, right? So, migrations, 1.8 to 2 million years ago, it's Homo erectus, and then 600 to 400 to 700, it varies depending on who you read. You have the Neanderthal, it really left Africa as Heidelbergensis and developed into Neanderthal. The Heidelbergensis that remained developed into Homo sapien. What's this? You probably all know it by now. They probably teach it in every course. But what is it that humans cannot do that every other mammal can do? Something physical, it's not speaking or anything, it's just a bodily function. Walk on all fours. Well, we can walk, walking upright. You can sometimes get your puppy dog to do it, but you have to have a lot of treats, but that's one. But I guess there are lots of answers, correct answers to that question. What I was looking for is breathe and swallow at the same time. Every other mammal can drink and breathe at the same time. We can't do that. Yikes, what's that about? Our larynx descended, probably to give us a greater range of vocalization. And when that happened, the anatomy of the throat changed. That's what we lost. That's why we choke on our food. In fact, that's how my mother passed. That's the price that we have paid for language. We're still kind of getting used to it as a species. We're still trying to get used to our neoteny with the snout shrunk. Our teeth are horrible. They don't fit, they're bulging out. We have the braces. You don't see squirrels with braces. They have perfect teeth. We are still rapidly changing and trying to make all the parts fit. So, what I call anatomically modern means that the larynx has descended. But did you know there was a time when each of you could actually swallow and breathe? In the first several weeks of life, you can still do that. That's why babies can't sing bass. Okay. Oh, man. Okay, the second Exodus, the southern route. Let's keep moving here. So this is the sort of a diagram of the different populations and changes in the hominids. 50,000 years ago, there was Neanderthal, Homo sapiens, and even some Homo erectus still walk the earth. Are these the original origins of the giants and the trolls under the bridge that we have in fairy tales? Are these remnants of tales taught around the fire? I don't know. Studies involving the archaea. Where does one draw the line of species? See, we're back again to species. Is it a thing? Species are defined by no longer having genetic exchange. You can't mate. But here we are mating with Neanderthal. So that means, by definition, Neanderthal is not a species. You see how arbitrary it all becomes. What is a species? It's however you define it. When did we become human? Here you see Homo erectus, that archaic form that didn't change much when it left. This is where it was found, there in the Middle East. There was a lot of variation. You look around the classroom, there's a lot of variation. You know, some people go, well, humans seem to be really lots of different types because we're humans and we can see the subtle differences. No, we really are really different from each other. We are a very diverse population. We haven't selected out yet in this new combination as we come together from the Neanderthal experience. As we have adapted to different regions of the earth, now we're coming together and exchanging genetic information again. We're becoming something new. There was a lot of genetic variations in this one cave of Homo erectus. When did man become anatomically modern? Was Neanderthal, could he swallow and breathe at the same time? Turned out he was like us. He could not. He had, the larynx did descend. Perhaps not quite as fully. Some of the reconstructions suggest that maybe he had a little trouble with ah. It was more like instead of ah. So he talked with an accent. Anatomically modern happened before Neanderthal and Homo sapiens split. So it happened while we were still Heidelbergensis. So that was, at least we had already become anatomically modern 600,000 years ago before Neanderthal broke off as Homo Heidelbergensis. How about the way we look now? Did Homo sapien continue to change? Yep. All the cold adaptation wasn't done by Neanderthal. Populations that live in cold climates continued to change. And so the genetic variation in our appearance and morphology is a complex feature that has been contributed to throughout our evolution. This illustrates the genetic evidence for the so-called bottlenecks. You see it in the Y chromosome, which of course is in the male. You see this big explosion with the bottleneck being represented by lack of diversity in this portion here, early lack of diversity, then an explosion into the different groups. The same kind of bottleneck appearance occurs in the female mitochondria. Of course, all mitochondria come from the female, right? You inherit your mother's mitochondria. And this just nice little illustration of the spread. So let's talk about another example of molecular biology, where molecular biology, we can study a particular mutation and see if we can link it to how we are distinguished from the more archaic humans. And that's what appears to be a transcription factor, something that it's not an enzyme, it seems to bind DNA and affect the way genes are expressed. It's called FOXP2. FOXP2, if you mutate it in humans today, you can look through populations and you don't mutate it, but you find people who have it mutated, and you'll see they have great difficulty with language. They can't construct sentences and things like that. If you look at it in birds and chimps, it's there. And birds, it can be, seems to be related to the ability to repeat sounds, to mimicry, learning of a language, FOXP2. So it's thought that changes that happened in the FOXP2 gene, because it's highly conserved, yet Homo sapiens has picked up some recent mutations, or the hominid line has picked up some mutations that you don't see in chimpanzees, for example. Maybe that's related to language. And of course, language is essential, a very kind of a hallmark trait that we have, capacity for higher language. Maybe higher cognitive processes leading to art, who knows. This is a FOXP phenotype. How about Neanderthal? Would he have FOXP2? Would he have the modern form or the original form? Well, that's a question that has excited and motivated those who are looking into the Neanderthal genome. And it was quite controversial about 5 years ago. And there's always the argument that you've contaminated with modern humans because we're constantly flaking off cells and stuff. So they pretty much prove that the Neanderthal genome has the modern FOXP2. So they got SIRGAP2, perfectly modern, FOXP2, perfectly modern. Let's look at brain anatomy. If you look here at the chimp or the bonobo, the closely related species, look at the brain and then expand the chimp brain to fit the size of our brain, you'll see the regions that you have to do the most expansion on. Some of the regions are in those regions that we know now are related to language and ability to imitate. There's a bump, Broca. There's a Broca's region on the left side only. If you look at the inside of a skull, you can see a little indentation on the left side that seems to be related to language. Is that why Homo sapiens has handedness, right-handed, left-handed, because the other things don't really have it? That's one of the theories. Again, that's one of the prices we have paid for language. We've had some specialization of the hemispheres. Broca's region. It's also a region where FOXP2 is expressed. Okay, so then let's get to the real stuff. Here's an exam question here. This will be on the exam, and it's in the homework so you can practice with it. We're going to introduce the concept of synonymous and non-synonymous mutations. You probably have studied them before, and they were called silent mutations. A synonymous mutation means that the resulting phenotype was the same, synonymous. No change, silent. So change at the nucleotide level. It changed the codon, the triplet codon, but it changed it into a version that still codes for the same amino acid. So it didn't result in an amino acid change, just a nucleotide change. Synonymous mutations happen all the time. They have essentially no effect because selection is not at the DNA level, but at the protein level. So synonymous mutations, very common. In fact, they appear to occur at a pretty constant rate depending on the species and the particular gene you're looking at. If you start picking up mutations that have no effect on the final protein, synonymous mutations, you can just count the number that have appeared over time and you can construct a molecular clock. And that is how we are able to put dates on some of these gene duplications and stuff that we've seen here in the slides. Molecular clocks. You look at the rate of picking up synonymous mutations. Well, what's the other kind? The kind of mutation at the DNA level that changes one base, but that one base changes into a different codon, meaning a different protein. Different amino acid is substituted now into the protein. Now you have the substrate for evolution to grind and work on. Most of the time, that's bad news. You change one amino acid, it's not as selective. Many proteins are highly conserved, just don't change for millions of years. So non-synonymous mutations, not the same, non-synonymous. These are the ones that evolution gets a chance to play with and say, is this going to be better or is it going to be worse like most of them? Non-synonymous. So what you see here is the non-synonymous mutations versus synonymous that have happened since the modern day, since the split. So that distance there in the mouse, this is FOXP2. From mouse to where the primates split off from mice, there were 127 synonymous mutations which had no effect. But only one time did a non-synonymous mutation take hold, become set, become fixed. Look at this. One happened between modern orangutans and the split with the gorillas, chimps, and us. So you see, nothing happened in this interval, nothing happened between gorilla and the chimps. But look what happened between when we split from the chimps and pygmy chimps. We picked up two. So how many changes from mouse? It was 1, 0, 0, 2, and those 2 happened in the hominid line since the split from chimps. Very suspicious. It's part of the evidence that these are important in making us what we are. So I'm going to ask you to read a little diagram like this. You'll get some practice in the homework. And just tell me how many non-synonymous mutations occurred between mouse and orangutan. How would you do that? You'd go from mouse to that branch point is 1. From the branch point to orangutan is another one, so that must be 2. And then from mouse to gorilla, there's 1 to that branch point, 0, 0, so just 1 from mouse to gorilla. And of course, to chimpanzee, from mouse to this branch point, there's 1, then 0, 0, 0, 0, just that 1. Homo sapien, there's the 1, and then that's 2, so it's a total of 3. It's not really complicated, but in this particular case, it illustrates the large degree of conservation in this transcription factor in mammals, and all of a sudden, it gets rambunctious when you come to hominids. And of course, Neanderthal had the modern version, which was a great disappointment to some people. They staked their careers on the fact that that's what made us different. And this is what I've just gone over. And then there's one last gene, Cincinnati, Tennessee, what? AP2. Connecticut, Tennessee, AP2. Connecticut, Tennessee, AP2. I always have to have stuff so I can remember it. Who could ever remember the CNTN-AP2 gene? So I always have to, Connecticut, Tennessee, AP2. I think there was a Confederate war general named A.P. Hill or somebody. So the AP kind of sticks in my head. Connecticut, Tennessee, AP2. This, if not the longest gene, it's right up there with the largest gene in the human genome. It just codes for a regular sized protein, but look how large it is. It's in the millions. I should have this. It's at least 1 million in terms of its length. It takes about 16 hours before the RNA polymerase can make an RNA copy of it. 2.3? 2.3 million. It is huge, as our president likes to say. Why is it so large? Because it has all these introns that are inserted in it that have to be processed out. Who thought up that? That seems like a totally illogical way to do anything, right? It's mostly junk, as far as we know. But here's the interesting thing. Notice it has a binding site for FOXP2. We don't know why. It's just an interesting aside. Oh yeah, it comes in here after I've humiliated myself. It comes to the next slide. Okay, so non-African populations have, on average, about 2% of the genome is Neanderthal. So that means non-Africans are about 98% Homo sapien. Great. And of course there were several populations of Neanderthal-like beings. There was one that is called Denisovian. Denisovian. That was in a cave here in middle Siberia. See the red dot? There was a cave that in 1747 there was an old hermit who lived way back in that cave, and he fancied himself as being Saint Denis. And he would make his parade through the town with his son. And so they named the cave of Saint Denis. And in that cave they found remains of an archaic hominid, similar to Neanderthal, but not the same as Neanderthal. Another archaic, coal-adapted archaic form like Neanderthal. And so it also contributed to the genome. So we've had Neanderthal, when Homo sapien encountered Neanderthal, which was coal-adapted from Europe, the non-African population absorbed some of the genes. You know, we have, each person that's non-African has about 2% on average. It varies from 1.8 to 4% Neanderthal or archaic human. But in terms of how much Neanderthal DNA is in the population of man, the family of man, the estimates range from 30% to, some say, 70% of Neanderthal genes are scattered among modern humans. But each of us individually, non-African, would have roughly 2%. What does this say? Oh, we don't want to read that. Well, it turns out I subscribe to 23andMe. And this is what I found when I opened up my app. It said, congratulations, you are in first place out of your family and friends. They're sequencing people I don't know. The 1,000 that look most like me in terms of DNA similarity, these are like fifth and sixth cousins I don't even know about. Of those 1,000 that are most like me, I have the most Neanderthal DNA. That's why they gave me the little prize there. I'm top, man, 284. So if you look over here, you'll see how Neanderthal DNA is distributed among my chromosomes. This is my 23andMe Neanderthal spread here. And one of the genes I got from one of my parents, has made me 1/10 of an inch shorter. It could have been somebody. So anyway, you're being taught by a caveman. When did man become behaviorally modern? We think it seemed to be about 40,000 years ago. People are saying it's related to art, as sort of advanced art in the caves. But was Neanderthal totally stupid? How about my great 1,000 times grandma? Was she really stupid? I don't know. I'm now looking differently at Neanderthal. It turns out Neanderthal developed some technology that Homo sapiens did not develop at that time. Neanderthal learned about pitch as an adhesive to hold the spear point onto the shaft before they wrapped it. Pitch. You know how you make pitch? You have to take 2 goose eggs. This is an old family recipe. One on top of the other. You drill a hole in the 2 so they're connected as 2 chambers, and you put birch bark in the top chamber, and then you cover it with mud and build a fire around it, and you keep the temperature at about 720 degrees Fahrenheit, and the birch bark coalesces in sort of a tar like pitch and then drips down into the bottom chamber. You have to do this in the absence of oxygen. Development of pitch was very advanced, I must say. So that's what my 2% did. When did it become behaviorally modern? That's still in debate. But it seemed that Homo sapien had higher cognitive abilities, but Neanderthal was not stupid by any means. So you'll need to know the difference on the exam between synonymous and non-synonymous mutations. You'll need to know about a molecular clock. So let's just think about a molecular clock a bit. These are synonymous mutations. What's the best place to set up a clock? If you were setting one up, would you put it in the middle of an important gene? Because as soon as you get mutations there, they're going to be selected against, right? So that's not a good place. You want something just going to go tick, 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 tick through the thousands of years, a molecular clock. You want it to be neutral from selection. And so it turns out one of the best places is to put it in a region that doesn't code for anything. And what has been found is in the mitochondria. There's about 1,000 nucleotides that's just a primer for DNA replication. And that, because it doesn't code for any protein, has become a very useful molecular clock. So the take home is you don't put it in regions of genes that are expressed as proteins. You put it in something that really doesn't function at the protein level. It's just nucleic acid, where there's no advantage or disadvantage of the mutation. Mitochondrial D-loop region would be correct answer. Whoa, look at this. Exam 1 alert. Are we still continuing to change? Has there been any recent selection? Of course. One of the most well documented is lactose intolerance. Why do people develop lactose intolerance? Isn't that nuts? What advantage could that be? oops, it turns out that the original condition is lactose intolerant. Anybody who can drink milk is a mutant. Hopefully that makes ice cream possible and it's a happy mutant. We're all happy mutants, some of us are not. The lactose ability to tolerate lactose happened in the transcription control region. Most of the genes that allow us to digest milk sugar, lactose, are only expressed when we need it in the first several months and years of juvenile development. But then they're not expressed. There's no need for them to be expressed. But in cultures that began to live around the cow at first was good for meat and then cheese, but the milk was no good. Why is cheese good and milk is no good? We put bacteria and yeast into milk and let the bacteria eat all the lactose. It doesn't have the lactose. And what was left, it turned it into protein, and that cheese became good, and we could survive through the hard periods if we could eat cheese. But if we could drink milk, we could save a lot of babies through the periods of starvation, and that became a selective advantage where cows were available, were part of the culture. So you see a lot of lactose ability to tolerate lactose in same places that you see the development of the cow. It arose independently in several places on Earth. So if you are lactose tolerant, that mutation seems to have been picked up in the last 6,000 years. That's kind of recent. Some of the other genes that are being selected upon, like they're all being selected upon, we just don't know what the result's going to be, are disease resistance. These are ones where it's life and death, and the chisel, the hammer of evolution, that chiseling out of a new species, that chisel is sharpest in those which affect disease. Let's take a 5-minute break, and we'll start in where this is all leading. We've talked about mutations and DNA and all that. The next topic is DNA replication, and then that goes into repair and how you fix it. And then the next section would be how genes are expressed in bacteria. And the last module of the course will be how genes are expressed in us, in eukaryotes. See you back here in 5 minutes.